When Jonathan was only 10 years old, his dad made a suggestion.
My father said, what if you rode your bike to Vermont?
240 miles.
Keep the bathroom fresh for whoever's got next.
Celebrate Women's History Month with the incredible women.
All the ads. Good day and welcome to MetPhys 2020, day 15, where we get to hear from Sophie Trefely about acyl-CoA metabolism and subcellular flux analyses. I've said this nearly every day, and I'm going to say it again. What we're building with metabolic physiology in isolation is not just a nice seminar series, but it really is a new model for inclusive global science. It's cost effective. The only costs those incurred by the White-Magara lab to get this to you on a large Zoom webinar setup. It's inclusive because everyone, anywhere and anytime can listen to these recordings and these talks, and I think that's really great. And then we can all be involved in the question period and ask as many questions as we like using the Twitter hashtag MetPhys2020. That's right. If you want to be part of the question period, use the hashtag MetPhys2020 within Twitter, and you'll be able to speak to the speakers that way. Even if you watch the talk in a year, you can use that at them, and they'll probably still reply to your questions. Also glad that it's green, that we're not causing anyone to travel, and that's nice for the environment. Once again, Cassidy has nailed it. She's there, I think, with the dreaded scoop monster, which she seemed to have tamed. It looks like a little purple furry thing, so we shouldn't be scared of him. Have a go, Cassie. Can we get 15 days in a row of photos in front of our computer? Last week, we saw the advent of a very new competition for MetPhys 2020. It seems the speakers are trying to take out the best background. I do think Isaac Harris took the lead. Gareth, I really appreciate your Aurora Borealis. It looked like you're in Seymour Skinner's kitchen. Anyone who liked The Simpsons will get that reference. But then Isaac, you came in with a meme and I really enjoy it. I don't know why they're all yelling at you, but even the cat seems a bit upset. Let's see if any other speakers will join the fun and throw in some fun and exciting backgrounds. I said it in the entry, the question period will be on Twitter. You can ask questions anywhere, anytime. Just use the hashtag MetPhys2020, and if you know the speaker's tag, you can @ them, and you can start asking questions as soon as you have them. And of course, when they finish speaking, they'll be able to get to those. If you don't have Twitter but you want to be involved, get Twitter. It's free. You do not need to have your name. You do not need to add friends. It's not Facebook. No one will know you're there. You can be a Twitter voyeur and just go to the search screen, type in MetPhys 2020 and click latest, and you will see all the questions people have asked and also the answers they've given. And that way you can at least listen to the conversation that's taking place after without having to out yourself. Although if you do, it's good that you'll be part of the community. I've said this, I think, every day, but we've got a lot of people to thank for these series. All the speakers who very rapidly agreed to take part. We're very grateful to have them part of this amazing lineup of talks. That's been extended to April 24 with these additional great speakers. After that, I'll need to take a hiatus, but I'm sure you will have all enjoyed those many days of talks. If you want to thank me personally, the way you can do that is to make a small donation to the Pathway to Stop Diabetes program at the American Diabetes Association. That is why I have my lab. They were able to support me. They're also supporting another speaker in the series, Celine Riera, who's going to come and tell us some fantastic work that she's doing on olfactory receptors and how they influence metabolism. And then tomorrow, I'm really excited about tomorrow's talk. Make sure you tune in again. Here we'll hear from Balazs Papagiannakopoulos from NYU, dissecting the role of metabolic rewiring in lung cancer. But right now, I'm going to pass it over to Sophie, and she's going to take it away. I'm going to give her the host controls and then get out of here. So enjoy, and I'll see you all tomorrow. Making you host, Sophie. Yes. Stopping my share. Stopping the video.
Okay. All right, so I'm assuming this is working, but I'm just gonna wait until I get confirmation from Phil. Not as yet. I wanted to thank Phil for organizing this great series and everyone that's tuned in. Okay, what was this? Waiting for screen to share. Okay, I need to share my screen. Okay, I think that should be running now. Okay, great. So I'm Sophie Trefely. I am a postdoc jointly in the lab of Katie Wellen at the University of Pennsylvania and Nate Snyder, who recently moved to Temple University in Philadelphia. I'd also like to thank the ADA, which has been really important in supporting me in a postdoctoral fellowship. So today I'm going to present my work on the analysis of metabolism at subcellular resolution. Okay, so this is a short outline of my talk. First, I will address our work on subcellular kinetic analysis using stable isotope tracing. And this is a study we recently published in Molecular Metabolism. Secondly, I'll explain SILAC-SF, which is a method that we have developed for subcellular acyl-CoA quantitation. And then I'll highlight two exciting findings from the application of these techniques regarding compartment-specific nutrient sensing and the regulation of metabolism within the nucleus. So everyone is in some sort of isolation right now, and many of you might be far from home and missing things from home. And something that I can't really decide if I miss from Sydney, where I'm from, are these creatures, long-finned eels. So these can be found sliding around in freshwater ponds all over the Sydney region. And these eels will live in the same pond for an average of 40 years. I assumed that the eels never went anywhere else. And I would never have drawn a connection between the eels in the local park and New Caledonia. But then I found out that they have a complicated life cycle where they transition from freshwater ponds to saltwater one time at the end of their life to breed. But they don't just go to the ocean off Sydney. Instead, they swim 1,300 miles to waters off New Caledonia to breed. And what's even more remarkable is that the tiny eels that are born in New Caledonia swim all the way back to Sydney to live. And this is known because they have been traced at various stages of their development, maturing as they get closer to Sydney waters. So the lesson from this is that tracing over space and time is essential for understanding the origin of eels. But the same principle applies to metabolites within cells. So metabolites also have complicated life cycles and are subject to many transitions, which are separated into different subcellular compartments. And this is very important because metabolites have different functions depending on their subcellular location. As an example, we can look at how glucose, metabolized in the mitochondria, finds its way into acetyl-CoA in the nucleus. Glucose derived from acetyl-CoA, glucose-derived acetyl-CoA feeds into the TCA cycle in the mitochondria, where it contributes to energy production. Importantly, acetyl-CoA cannot be directly transported across the mitochondrial membrane. Instead, citrate is exported from the mitochondria and cleaved by acyl-CoA to produce acetyl-CoA cytosol. And this has many functions in the cytosol, including as a substrate for fatty acid synthesis. acyl-CoA can also produce acetyl-CoA in the nucleus. And this acetyl-CoA has functions in influencing epigenetic marks through histone acetylation. Additionally, multiple substrates can contribute to metabolites through compartment-specific pathways. For example, acetate can be directly converted to acetyl-CoA both in the cytosol and in the nucleus. So you can see that if you want to understand how metabolites are regulated in their compartment-specific functions, you need to be able to trace their origin in space and time. Stable isotope tracing can be used to visualize metabolic pathways in time. So by adding a labeled substrate to cells, such as 13C glutamine represented here, we can watch as these 13C carbons are incorporated into downstream metabolites using mass spectrometry. So in the case of glutamine, we can see as carbons from glutamine are passed into alpha-ketoglutarate and subsequently into succinyl-CoA with an increasing half-time of incorporation as they progress down the pathway. But this technique in whole cells does not tell us about the dynamic relationship between metabolites in different compartments or potential alternative metabolic pathways. In a standard stable isotope tracing experiment in whole cells, cells are incubated over various time points with heavy isotope labeled substrate, glutamine shown here. After incubation, the media is rapidly removed and metabolites are directly extracted for analysis by mass spec. Importantly, we always include a unlabeled control in parallel, and this is used to determine the background 13C levels. So we can then calculate the 13C enrichment of metabolites of interest in our tracing cells. Subcellular metabolite tracing is a major challenge in metabolomics because it requires specifically measuring metabolism within each compartment. Previous work in this area has used computational approaches to analyze and deconvolute whole cell data. This approach has generated some valuable insights into metabolic pathways, but it's limited because it relies on assumptions about metabolic pathways, and it's restricted to annotated pathways. Physical fractionation has the potential to generate new insights into metabolism, but has suffered from issues of reproducibility due to metabolite instability. This has led to people avoiding these experiments. These issues have led to people avoiding these experiments altogether because fractionation to separate compartments is a disruptive process with the potential to introduce multiple artifacts and errors in interpretation. So these experiments can generate data that may not readily be validatable. But we wanted to explore the possibilities of this technique, and to do this, we dealt with the issue of potential artifacts head on. Firstly, we performed the same kind of tracing experiment as we would for whole cells and incubated cells over a time course with tracer, but instead of directly extracting metabolites after incubation as we would here, we added fractionation buffer and performed several steps to separate out different compartments before extracting them separately. So we're quite excited about our initial results from this approach. They seem promising in that we were able to detect distinct kinetics in different compartments. So here is shown the data for succinyl-CoA labeling from glutamine over time. And you can see that mitochondria in pink is labeled really fast, whereas the cytosol in green and the whole cell lysate in gray are much slower. They're 15-minute half-time compared to 4 minutes in the mitochondria. But succinyl-CoA is primarily a mitochondrial metabolite, and therefore we'd expect the whole cell lysate to reflect the mitochondria more closely. So the disconnect between the 15-minute whole cell half-time and the 4-minute mitochondrial half-time seemed mathematically impossible. And this made us suspicious, so we wanted to investigate if metabolism during processing might have influenced this result. To directly test the extent of artifactual metabolism, we designed a post-labeling scheme where we added isotope tracer to cold cells in the fractionation buffer instead of in the incubation media. This way we can detect artifactual metabolism occurring from the point of cell harvest. Part of this strategy was to take a whole cell lysate aliquot immediately after scraping the cells. So this is within 30 seconds of placing those cells on ice. The second whole cell lysate aliquot was taken after homogenization to compare. When we added the 13C glutamine tracer post-harvest, we saw significant label incorporation in the mitochondrial fraction, whereas we see no labeling in the cytosolic fraction. This indicates that post-harvest metabolism reflects compartment-specific metabolic capacity, since the mitochondria is a major site for glutamine metabolism. But when we added 13C labeled alpha-ketoglutarate, we saw the incorporation into succinyl-CoA in all fractions, and this was an overwhelming amount of incorporation. So over 50% of the total succinyl-CoA pool was labeled by alpha-ketoglutarate in the whole cell lysate number 1. So that's within 30 seconds of adding the tracer to cells, cold on ice. So alpha-ketoglutarate can be directly converted into succinyl-CoA. This is just one metabolic step, whereas glutamine requires an additional 2 steps in order to become succinyl-CoA. So to assess the extent of post-harvest metabolism, we looked further at the labeling of more distal metabolites. And here you can see that we actually detect no labeling into distal metabolites, acetyl-CoA or propanol-CoA, from either alpha-ketoglutarate or glutamine. So this indicates that post-harvest metabolism is limited to proximal metabolites in metabolic pathways. So the compartment-specific nature of the post-harvest metabolism that we detected indicated to us that it's likely enzyme-dependent. And we tested this by tracing 13C acetate into acetyl-CoA, a reaction that's catalyzed by the enzyme ACSS2. In the Wellham lab, we have ACLY knockout fibroblasts that show extremely high expression of ACSS2. So as you can see here. But we also have ACSS2 knockout fibroblasts with no ACSS2 expression and normal MEF cells with an intermediate level of expression. So we can use these cell lines to titrate the amount of ACSS2 and then assess the degree of post-harvest metabolism from acetate. And when we did this, we can see that the higher the level of ACSS2 expression, the higher the degree of post-harvest acetate conversion to acetyl-CoA we can detect. So this indicates to us that post-harvest metabolism is enzyme dependent. So to summarize this section, post-harvest metabolism occurs rapidly within seconds of cell harvest. It's enzyme dependent, it's compartment specific, and it's limited to proximal metabolites in metabolic pathways. Since we know that post-harvest metabolism occurs unavoidably during fractionation, we developed a strategy to account and correct for it. Our strategy is to take advantage of post-harvest labeling. By adding the relevant tracer to the fractionation buffer of both pre-labeled and post-label cells in parallel, we can use the control to normalize against and account for metabolism occurring post-harvest. We found that this strategy could transform our kinetic data. Shown here on the left is the uncorrected succinyl-CoA data from earlier, and on the right is corrected with post-labeling. You can see that the half-time for mitochondrial labeling from glutamine is now consistent with that of the whole cell lysate, as we would expect with succinyl-CoA being predominantly mitochondrial. Post-harvest metabolism can be an issue not only for subcellular fractionation experiments, but really for any kind of metabolite analysis. In this experiment, cells were incubated with 13C labeled glucose, and then the whole cells were directly extracted into trichloroacetic acid or into 80-20 methanol water. And the results from these two different extraction solvents is exactly the same, validating the direct extraction approach. When we introduced additional processing steps before extraction, including PBS wash and scraping cells into media, followed by centrifugation before extraction, we get very different results, indicating artifactual post-harvest metabolism occurring with extra processing. So having established that direct extraction into solvent is the gold standard for whole cells, we use this to help validate our post-label correction strategy. Here I've depicted the TCA cycle in linear form, starting with glutamine, and then progressing to alpha-ketoglutarate through succinyl-CoA, succinate, malate, and citrate. And these green circles represent the 13C carbons as they're passed through the pathway. So these green triangles show the half-time of glutamine labeling in whole cells with direct extraction. So that's our gold standard extraction, looking at the TCA cycle. And you can see that the half-time increases as we progress through the TCA cycle. We can then compare this to our succinyl-CoA data from fractionation with and without post-labeling. So if we look at the mitochondria from the uncorrected data, the half-life is really an outlier. Whereas with post-label correction, this mitochondrial half-life is aligned with the rest of the TCA cycle. So this helped us to validate our approach, and we think that this correction strategy will allow us to perform subcellular tracing experiments to determine the dynamic interactions between metabolic pathways in different compartments, and has the potential to uncover new insights into metabolic regulation and potentially signaling. So I'll now compartment compartmentalization of metabolism means that subcellular concentration of metabolites can dictate their function. But to achieve robust subcellular quantitation of metabolites requires different approaches. The family of metabolites that we are particularly interested in are the acyl-CoA thioesters. Acyl-CoAs form a large and diverse family of metabolites that are intermediates in multiple metabolic pathways, making them ideal indicators of metabolic status. But they can have different functions in different cellular compartments. As I showed earlier, mitochondrial acetyl-CoA is generated through a number of catabolic pathways and contributes to energy production. Cytosolic acetyl-CoA, on the other hand, contributes to anabolic processes, including cholesterol and fatty acid synthesis. Acyl-CoAs also have the potential to act as substrates for protein acylation in all compartments in a manner that's dependent upon their concentration in that specific compartment. These functional outcomes highlight the importance of subcellular quantitative metabolomics. Recent progress in subcellular metabolomics has included the development of an immunoprecipitation method for the rapid isolation of target organelles, followed by analysis by LC-MS. This method has some limitations, which we set out to address in our approach to subcellular acyl-CoA quantitation. I won't go into all of these, but the first and most important to note is that internal standards were added after fractionation. So internal standards are an important tool in mass spec analyses. Appropriate internal standards are essential for accurate quantitation of acyl-CoAs because they are particularly unstable molecules. We use SILAC so that's stable isotope labeling of essential nutrients in cell culture, to generate heavy labeled internal standards for acyl-CoAs. And this is a similar approach to SILAC used for amino acid labeling in proteomics. So SILAC internal standards are generated in mammalian cells by passaging cells over multiple generations in media where vitamin B5 is replaced with heavy labeled vitamin B5. And this is incorporated directly into the CoA backbone of acyl-CoAs. So for routine whole cell measurements by LC-MS, we use SILAC acyl-CoA extracts to spike into samples in equal amounts before processing, so that the ratio of the analyte to the internal standard can be used to compare the relative quantity between samples. In collaborations between the Wellen and Snyder labs, we've applied SILAC internal standards to quantify acyl-CoAs in whole cells and tissues across multiple biological scenarios. This has allowed us to determine the biological functions of acetyl-CoA metabolism in diet and metabolic disease and in cancer. But we wanted to take this SILAC internal standard one step further and take advantage of the fact that it can exist in a whole cell living form. We developed SILAC-SF, SILAC subcellular fractionation, in which the SILAC internal standard is introduced at the beginning of processing before fractionation. First, we scrape our SILAC cells into fractionation buffer, and then we add equal amounts of these cells to each sample, which can be cells or tissues. This means that the internal standard is present as a whole cell before any processing for fractionation occurs. In that way, the internal standards account for sample loss, chemical changes, metabolism, and processing errors that happen from the point of cell harvest. After SILAC addition, the cells are fractionated to separate different compartments. Importantly, the internal standard will be present in every relevant compartment. Each fraction is then analyzed by LC-MS. And the relative heavy to light ratios can be compared between samples within each compartment. So So the SILAC-SF approach is compatible with any fractionation procedure. And the data I will show you today was generated using differential centrifugation methods in which we isolate mitochondria and cytosol simultaneously. So we applied SILAC-SF to quantify acyl-CoAs in mitochondria and cytosol in a variety of cells and tissues. Shown here is data from brown adipocytes and MEF cells. The acyl-CoA profiles are distinct for different compartments across different cell types. Notably, acetyl-CoA and succinyl-CoA are the most abundant acyl-CoA species on a whole cell level. Whilst in the mitochondria, succinyl-CoA becomes the dominant species, and in the cytosol, acetyl-CoA becomes more dominant. We are also able to show the same pattern in human heart tissue, with a relative enrichment of succinyl-CoA in the mitochondria and acetyl-CoA in the cytosol, shown here with Western blots to validate enrichment of compartment-specific proteins. So I've shown that we can generate consistent and reproducible data for compartment-specific acyl-CoA quantitation. But we needed to validate that our approach can detect biologically relevant perturbations with a predictable outcome. So we leveraged a well-characterized metabolic phenomenon, the induction of reductive carboxylation upon hypoxia. So under hypoxia, the TCA cycle is reversed such that carbons from glutamine are directed to citrate production and away from succinyl-CoA production. Since hypoxia diverts metabolism away from succinyl-CoA, we predicted to see less mitochondrial succinyl-CoA under hypoxia. And that's what we saw when we did our SILAC-SF analysis. So succinyl-CoA was reduced in the whole cell level and significantly in the mitochondrial compartment. But cytosolic levels of succinyl-CoA remained unchanged, indicating a separately regulated metabolic pool. When we look at the effects of hypoxia on a panel of short-chain acyl-CoAs we quantified, it's clear that, as expected, hypoxia preferentially impacts mitochondrial pools, indicated in pink at the extreme ends. And that's compared to the cytosol and whole cell changes, which are much less impacted. So we wanted to apply this SILAC-SF technique to investigate acetyl-CoA metabolism. And since the relationship between acetyl-CoA concentration and histone acetylation is a specific example of the importance of cellular metabolite, subcellular metabolite local concentration. So as I mentioned at the beginning, acyl-CoA cleaves citrate in the nuclear and cytosolic compartment to produce acetyl-CoA. And deletion of acyl-CoA limits the supply of acetyl-CoA in these compartments and thereby limits histone acetylation. And this affects gene expression and impacts multiple physiological processes important in disease, which have been investigated by the Well Lab and many others. These include cancer and cell differentiation. But metabolism is adaptable, and acyl-CoA knockout cells compensate for the loss of nuclear cytosolic acetyl-CoA by upregulating ACSS2 and using acetate as an alternative substrate. These cells are dependent on exogenous acetate, and they eventually die in the absence of acetate. They're also dependent on acetate for histone acetylation. You can see that histone acetylation levels can be rescued to the level of control cells with the addition of a very high acetate concentration of 1 millimolar. But the cells, despite histone acetylation being rescued at this high concentration, the cells are viable and will grow happily in a more physiological 0.1 millimolar acetate concentration. Given the relationship between acetyl-CoA concentration and histone acetylation, we thought that as we titrate up the acetate concentration, we should see a corresponding increase in acetyl-CoA concentration. We investigated this in whole cells using our gold standard direct extraction method. And whilst acetyl-CoA levels increase from 0 acetate to physiological 0.1 millimolar acetate, as we would expect, acetyl-CoA does not respond to further increases in acetate. We also looked at the response of a panel of other short-chain acyl-CoAs. And one metabolite that really stood out was HMG-CoA, which responds in a stepwise manner to acetate titration, which is what we expected acetyl-CoA to look like. This disconnect between acetyl-CoA and histone acetylation data made us think that maybe this could be explained by an accumulation of acetyl-CoA sequestered away from the nucleus in the mitochondrial compartment. So we performed SILAC-SF to compare the acetyl-CoA levels with the 0.1 and 1 millimolar acetate addition, specifically in the mitochondrial and the cytosolic compartments. And we saw the same as for the direct extraction. There was no change in either compartment. That was what was very interesting though, was that we did see a significant response in cytosolic HMG-CoA to acetate dose. So acetate, HMG-CoA is a downstream product of acetyl-CoA metabolism in the cytosol and supplies the mevalonate pathway, which has multiple functions and roles, including in cholesterol synthesis and in ubiquinone synthesis. So HMG-CoA could potentially be tuning this mevalonate pathway according to acetate availability. To summarize what we found in these experiments, acetate-dependent ACOI knockout cells have reduced acetyl-CoA levels and are unviable without acetate. At physiological 0.1 millimolar acetate levels, cells are viable and cytosolic HMG-CoA levels respond. Whereas supraphysiological acetate leads to further increases in HMG-CoA with the potential to influence biological functions through the mevalonate pathway. And histone acetylation in the nucleus is only increased in this high acetate concentration. So this implies that there could be separate regulation of the nuclear and cytosolic compartments in terms of acyl-CoA metabolism, despite